ফুল স্ক্রিন করো।
Done.
Allow the full screen PowerPoint.
Sangachadvam Sambadadvam Sangvo Manansi Janata Samano Mantra Samiti Samani Samanam Mana Sahajita Mesha Sangachadvam Sambadadvam Sangvumanansi Janata Samani Vayakuti Samana Hridayaniva Samani Vayakuti Samana Hridayaniva Samanamastu Mano Yathava Susahasati Sangachadvam Sambadadvam Sangvumanansi Janata.
Namaskar.
Pranam. Very good
Sushagatam.
evening to everybody.
A very good evening to all of you. Welcome to the two-day international webinar organized by the Department of Botany and the Department of Zoology in association with IQSC of Charuchandra College, Kolkata. This evening and tomorrow, our topic of the international webinar is sustainability amidst the COVID-19 pandemic. We are extremely happy that a lot of faculty members, researchers, and students have joined us through the YouTube platform. I am pleased to announce this evening on this webinar panel, we have among us Professor Anuradha Ghosh, teacher in charge of Charuchandra College, Dr. Suparna Sen, IQSC coordinator of Charuchandra College, Dr. Kaushiki Chakraborty, my joint convener, and two very distinguished invited experts, Dr. Subharti Das, research scientist and co-founder of Bioli LLC, who is joining us just from the opposite side of the globe, from California, United States. Yes, it's early morning, so a very good morning to her. And Dr. Ratnadeep Ganguly, another speaker for today, consultant histopathologist from Coventry, England, will be joining us soon. And we are very happy that Dr. Sandeep Poddar, Senior Research Director and Executive Editor, Publications of Lincoln University College, Malaysia, has also joined us for this inaugural session. He is our invited speaker for tomorrow's another very important session. Now, I would like to request Professor Anuradha Ghosh Madam, the teacher in charge of Charuchandra College, to deliver her inaugural address. Over to Professor Ghosh, madam.
Well, good evening from Calcutta to all of you who are from all over the world. And well, I welcome you on behalf of Charuchandra College. And as we all know, we are going through a very unprecedented situation in this pandemic thing that is going on in this world. Well, we are really in a state of sort of flux. We are feeling very insecure. We are feeling restless. We do not know what to do, how to go about. So we really need some help to sustain ourselves now. So I think the speakers who would be speaking on this aspect will enlighten us, enrich us with their erudition, with their their advice, and obviously we will remain enriched after that. And now it is my honor to say that this webinar is open.
Thank you, Professor Anuradha Ghosh, our Honorable Teacher in Chair, Charuchandra College. Thank you for your inaugural speech. Now, I would like to invite Dr. Suparna Sen, Coordinator IQSC, Charuchandra College, to deliver keynote address. Over to Dr. Suparna Sen.
Thank you, Kaushiki. First of all, on behalf of IQSC Kolkata, Charuchandra College, Kolkata, I would like to extend a very warm welcome to all our esteemed speakers. This webinar will continue for 2 days, today and tomorrow. Today, our esteemed speakers are Dr. Subharti Das and Dr. Ratnadeep Ganguly, who will speak on a very important topic, sustainability amidst the COVID-19 pandemic. Tomorrow, this webinar will continue, where Professor Somendra Saha and Srilekha Saha will be speaking on the health and well-being as a function of sustainability amidst the COVID-19 pandemic, and Dr. Sandeep Poddar on impact of COVID-19 on health, environment, and economic sustainability. All these topics, all these issues are very important for us. We are all directly concerned with this. And as our teacher in charge, madam, has pointed out, that we are all rather confused. We do not know how to come out of this pandemic or how to deal with it. We are also very much interested in this new normal style of life, because this virus is not going to leave us very soon. We have to cope with it. We have to deal with it. So we are all very eagerly awaiting to hear what our esteemed speakers would have to say. And so without wasting much more time, I would request Professor Subharti Das and Dr. Ratnadeep Ganguly today to make their speeches, which will surely enlighten us and help us to overcome these difficult and unprecedented times. Thank you.
Thank you, Dr. Sen, madam. Now we are ready for the technical session 1. As Dr. Sen, madam is speaking, telling us about, the several months have gone, but the pandemic is not yet over. We are still in the middle of this COVID-19 pandemic. Since the very beginning of this dreadful 2020 year, we are fighting against the deadly SARS-CoV-2 virus. In countries like China, it had started even earlier. But in this middle of this August, the graphs of infectivity and mortality are still rising. Too much administrative confusion, contradictory statements, unscientific remedies, economic slowdown, health system inadequacy, mental agony, all these are going on. It shows how helpless we are in the hands of nature. Isn't it?
Exactly. Indeed. Really, we are helpless. So what to do? how to get rid of this situation. We have to think and rethink about different parameters of sustainable development. As we all know, sustainability or sustainable development may be defined as development that meets the needs of the present without compromising the ability of future generations to meet their own needs. The COVID 19 outbreak has shown the world is unprepared to react promptly to global health threats. This unpreparedness does not only apply to the management of pandemics, but also to the prevention of irreversible consequences of climate change. While we are facing pandemic, a new thinking emerges to manage nature and its resources. So far, our policy response to the COVID 19 outbreak has been based on the principle of safe living, along with the principle of sustainable development for a safe future. Due to the highly contagious nature of the disease, people have been left with no option but to stay at home and take multiple precautions at a personal level. We are facing an extraordinarily tough situation that has compelled us to think how to manage nature and its resources differently to avoid pandemics like COVID 19. At a time, it is imperative to revisit the established principle of sustainability for the purpose of keeping focus on safe living, leading to health security for the people. The principle of sustainable development needs to be strengthened adequately, even by adding new management parameters. However, the critical question remains that what needs to be prioritized or added to the existing principle of sustainable development. So, to discuss about different parameters of sustainable development, our esteemed speakers, they have reached here from different parts of the globe to enlighten us. Now we will start our technical session 1. Our first speaker is Dr. Subharti Das from USA. I would like to invite Dr. Suman Das to introduce Dr. Subharti Das. Over to Dr. Suman Das.
Thank you. Dr. Subharti Das is biomedical research scientist and entrepreneur, co-founder of BioLi LLC in California, USA. She has worked as a scientist in the US's first mass immunity testing program by Stanford University, and presently she is working on a very significant multinational collaborative project reviewing the accessible and affordable diagnostic methods and surveillance tactics for COVID-19. After her master's in botany from University of Calcutta, she did her PhD from University of South Carolina. After doctorate, she did her postdoctoral works in University of Massachusetts Medical School, University of California, Davis, and eventually in Stanford University. She has expertise in immunomolecular biology, gastroenterology, hepatology, toxicology, and all these. Author of many peer-reviewed publications in top-tier journals, she received many research grants and awards, including Distinguished Graduate Student Award and Breakthrough Graduate Scholar Award from the University of South Carolina. Her topic today is sustainable pandemics surveillance and mass diagnosis, living the new normal together. Over to Dr. Subharti Das. Hello, excuse me, Dr. Das. We can't hear you. It was okay just earlier. Dr. Chakraborty? Is the microphone jack is Is the microphone jack working?
Can you hear me now?
Yes, yes, yes. Audible.
Okay. Okay. It showed somehow it clashed with the host unmuting and all. It showed host has to unmute, but then I could unmute myself. But anyways, so thank you first of all for inviting me to this very timely initiative. And when Dr. Shuman Das reached out to me about speaking on this webinar, I definitely could not deny because it's very special. It's Kolkata, my hometown. And in addition to being a scientist, in addition to being a researcher and doing my research work for a decade now in the USA, I also have family and parents back in Kolkata. I'm a daughter and a daughter-in-law. So in the midst of this unprecedented pandemic, it's kind of a bridge, or I would say a split between trying to make our relatives, our family, and everybody back there who are from a non-scientific background understand effectively what are the implications of this new normal and how to deal with it, um, as well as, of course, generating new knowledge and new research and enlightening the students, the young generation, and the science-minded people. So with that in mind, I have come up with a presentation which is, I think, very much aligned with the topic of today's webinar, which would have two main portions. That would be definitely the mass diagnosis and surveillance, how to do that scientifically, how to do that policy-wise. And also there would be another portion where we, the people, all of us can do our bit in maintaining our personal protection or hygiene, and at a social level we can adapt to this new normal. So with that, without much ado, I would like to share my PowerPoint presentation. Maybe one. Okay. Can Dr. Shubhan Das or Dr. Chakrabarti, can any of you confirm whether you can see my PowerPoint presentation as a full screen mode?
Okay, proceed, please.
Okay, all right. All right.
Okay.
So here's my topic: sustainable pandemic surveillance and mass diagnosis, living the new normal together. I would definitely like to acknowledge the research at Stanford University with which I was involved. I'll be talking in detail about that experience and how that helped in also, the evolution of myself as a scientist in the need of the community or the society in times of unprecedented pandemic as well, and also independent research and consultations, SWIB meetings with global collaborators. And I just want you to remember that right now, unlike times before where there has been different disease outbreaks or a lot of other kinds of contagious epidemics all over the world in the history as we know, this is very different. All of us, most of us around the globe are affected mentally, psychologically, financially, definitely health-wise. This is just a disclaimer that this is the going into the intricacies of how the disease is spread, and going into the scientific details is just for the purpose of generating knowledge. And also this is very much evolving, so the data and graphs can change very soon. So I know this is about mass diagnosis, surveillance, and sustainability, but I'll very briefly touch on the predicted origin and coronaviral genomics, because I know I was told that there are several undergraduate and postgraduate students and professors in the audience who would be interested in knowing or sometimes being reassured about the right origin and how the prognosis or the etiology of the disease right now. That being said, this is probably one of those unprecedented times for us researchers and presenters where the audience, unlike many other times, probably knows equally or sometimes more than the presenter. So it's easy to communicate, but it's difficult. With that, let me delve into the origin and the genomics. So as of now, scientifically, it has been reported that the coronavirus, now nomenclatured as COVID-19, is novel in the human race, but it always or existed for a long time in the environment. It's not new. What happened is due to certain interventions, certain changes, and as the speakers before me, the conveners of the conference were also touching upon, that due to different kind of interventions with the environment by the human race, who are known as the most intelligent race of all, certain changes, certain devastating changes, certain unwanted things have been happening. Possibly one of those are the intervention, recombination of this virus in a zoonotic host, and then invasion into the human race. So far, with a genomic study, it has been found that there is around 96% of whole genome sequence similarity to the rat G13 virus in bats. There is a 99% genomic concordance with the virus in pangolins, but that is only a particular region of the virus, which is the S encoding region. So from the studies, it has been implicated that probably pangolins were the most likely host before human transmission, and a very significant or small recombination having a significant effect has happened in the SARS-CoV-2 genome. We don't know for sure whether that recombination happened in the bats or the pangolins or any other part of the environment, but definitely unwanted mixing happened, and that's why it invaded the human race. So what are the challenges in controlling this COVID-19 pandemic? What is all this brouhaha about? Why isn't not like any other disease or any other flu, like some people has been telling that, just pop a pill or drink more water or take some immunity boosting supplements and you will be fine. A few months of our experience has shown that it is not so easy. Okay, a few of the factors which sets this disease or this pandemic or the COVID-19 apart are that it uses the population density to its advantage to run like a very aggressive community transmissible virus. So it transmits very easily. And we all know now that that can be transmitted, the disease can be transmitted or being infected. A person can be infected from an already infected host through droplet transmission. If they are more than 5 micrometers in diameter, when you sneeze or you cough or you speak loudly, then that usually goes and affects people who are within 6 feet distance. That's why we have been hearing a lot that stay 6 feet apart, do social distancing. But also right now with recent research, it has been shown that aerosols or the particles which are less than 5 micrometer in diameter, they can also carry the infections further than 6 feet distance, which is very worrisome. The other factor is that these have a long viability. The viable particles can stay alive for very long on surfaces like plastic or paper, glass, gloves, steel, metal, any other kind of things like aluminum. Those are called fomites. Usually what happened is because the other kinds of virus which were or very common in the human race before did not have that long susceptibility or viability. So they were not so much contagious and did not affect so many people so easily. Also, once it enters the human body or the host, it's an aggressive inflammatory progression that is being shown, very aggressive, very fast, usually. Now, the human body or immune system is very varied. So hosts might have varied reactions, and we have seen that. Also, there are two factors going on here. I heard one of the scientists describing this as a wild virus which was not introduced to the human race, and slowly as it is undergoing mutation and slowly as different people are being infected time and again, they are growing certain amount of antibodies or some kind, at least partial immunity against this. So the infectivity is high, but the mortality rate worldwide are at least going down. If you look at the whole picture, I know it's very weird or strange to hear because you see people dying left and right and being affected. But statistically, it has been shown that from the initial months, mortality has gone down a lot. But that being said, we cannot rest easy because the virus is very mutable. And although we are looking for therapies and diagnostics and sustainable vaccinations, still we have to keep the mutation and the changing nature of the virus in mind. Right now, what's the modus operandi of SARS-CoV-2? Once again, I don't want to spend a lot of time because I think most of the people now, whether you are from humanities or science or any other kind of field or of interest, you have delved into the details and you know that it enters your body through the respiratory system, it goes to your lungs, and then affects the small sacs or the baglets that are present in your lungs called alveoli. So because when there is healthy breathing, usually what happens is through the layer that's between the tissue layer that's between the alveoli sac and the blood channels or the capillaries, the oxygen passes or permeates through that, and it's being carried to the heart and rest of the organs of the body for proper oxygenation. During an attack by the SARS-CoV-2 virus, which is the causal pathogen of COVID-19, it goes and there is an inflammation, and the inflammatory cells pour into the lungs. The membrane thickens, and so the alveoli are suffocated, sometimes filled with mucus and fluid. So oxygen can't reach the blood, and without oxygen, the organs fail. The immune system goes into an overdrive. In most cases, or at least in some, there is a cytokine storm, which means the inflammatory molecules. And as I said, the organs are oxygen deprived. There are clots in the blood vessels. So as you can imagine, as you can understand from this, it has an all-pervasive effect in your body. It's very difficult to survive if such an aggressive progression goes on. You have breathing trouble or respiratory distress. There are several other symptoms that's been reported now, that you lose your sense of smell, there is olfactory dysfunction. It affects your nervous system, sometimes your brain, without showing any other kind of effect at any other parts of your body. The very important factor for SARS-CoV-2 is that it is an aggressive and tactful invader. That is, we have all been hearing these terms, comorbidities and pre-existing conditions. So it uses the already existing chronic or insignificant amount, which were not the diseases, which were not bothering you that much in your lifestyle. You were just maintaining those, using some medications or something. When that happens, that you have a little pre-existing condition, then SARS-CoV-2 utilizes it to its advantage. So when there is hypertension or diabetes, chronic kidney disease, or any other kind of heart condition, then definitely SARS-CoV-2 doesn't let you rest easy. And also it has been shown that recently it generates diabetes in people who did not have it before. That is, SARS-CoV-2 infection does incite diabetes. So that was kind of about the etiology and prognosis a little bit. And now I would like for specifically the young people out there who want to go into the field of science to look into this beautiful, well, when I say beautiful, that once again is from a scientist's perspective, but definitely these are images of our very grave invader of the human race right now. These are electron micrographs. On the top, you can see some images which were taken from the coronavirus isolates of the first American case of COVID-19. The blue particles, they have been artificially colorized to make them stand out, but those are tissue isolates. And that's how the coronavirus is spread inside our tissue. The second one at the bottom and the big one on the right, they are of cultured cells, which were again used to show coronavirus isolates from patients. And when they are cultured in a lab, sometimes in Vero cells or other kinds of cells, this is how they look under scanning electron microscope. On the right, you can see a big apoptotic cell. The yellow particles are the coronavirus particles, or the SARS-CoV-2 particles, and you can see what they do to a normal cell so that it becomes apoptotic. So let's look at the anatomy of a killer. And once again, I do not want to delve into a lot of details about this and go to our topic, but I want to mention the different kinds of protein present and also the RNA, because that is important to understand the mass diagnostics that I'll be mentioning afterwards. So we kind of know this is probably the most circulated, the image or the picture, different variations of this one now throughout the internet, unfortunately. But to look into the protein particles present here, it consists of an envelope protein, a membrane protein M, and a spike protein, which is very important, the spike protein, because that is used by the SARS-CoV-2 virus to attach itself with a receptor in the host cell, which is called the ACE2 receptor, ACE2 receptor, and then push through its whole body inside of a cell. Then the nucleic acid particles or the RNA particles are released there, and we'll see in the next slide whatever happens next. So when CoV binds with the ACE2 on the cell surface receptors of any kind of cells in your body, specifically in the respiratory tract, that's where it first attacks. It mediates, the receptor fuses with the spike protein, and then the viral RNA is released into the host cell cytoplasm after it enters the cell, where the viral nucleoproteins uncoat. Later, it's translated to produce the unglycosylated proteins, and they're trafficked through the Golgi body where they're glycosylated. And there is an assembly going on. So basically, we know what viruses do. It uses the host's mechanism of assembly and synthesis. Once again, it's a tactful invader, any virus is. So this uses to its own advantage and produces new particles, and the vesicles fuse with the cell membrane, and then they release the CoV virions into the lumen of the cells. So why this is important, the entry point, is because the antibodies that I'll be talking about, they neutralize, bind with the spike proteins and neutralize them, sometimes suboptimally, of course, but mostly completely, and that prevents the entry of this virus, that prevents the fusion of the virus with the ACE2 receptor, and hence the entry of the virus into the cell. If it cannot effectively enter the cell, the whole process of replication, synthesis, and whatever goes on after that, that's attacking all the different parts of the body, is blocked. So now going to the main portion of mass diagnostic tools and sustainable approach. Why is a reliable diagnosis so difficult to reach? Why the companies, the universities, research institutes, the regulatory bodies, and scientists all over the world, along definitely with the healthcare workers and the doctors, they are brainstorming, they're doing a lot of research, and yet cannot come up with a proper diagnostic or surveillance tool to fight this pandemic and control it so far. To understand that, we have to understand that there are several criteria that needs to be fulfilled. First of all, it needs to be fast, that is rapid, because it's an aggressive invader, remember, and it's highly transmissible in the community. It has to be accurate or near accurate. Of course, we cannot afford to have false positives or negatives a lot. That would lead to a lot of confusion. So on that front, there are two parameters. It has to be sensitive and it has to be specific. Sensitive meaning that even if there are very low amounts of virus or very low infection present, it's still the diagnostic method or the surveillance tool or the screening tool, it should identify that very easily. It has to be specific, which is very much essential for the antibody-based diagnosis and screening or surveillance method, because we know that the human body produces antibodies against a lot of pathogens, so it has to be specific against the coronavirus antibodies or the antigens. And very, very importantly, it has to be executable in low resource setting, because there are only very less percentages of laboratories or clinics or highly trained personnel in the institutes who can carry out very sophisticated methods like the real-time PCRs or proper ELISAs to detect the antibodies or the nucleic acid or the RNA. So that cannot be a practicable approach when a pandemic is aggressively invading people all around the globe. It has to be executable in a low resource setting. So making that accuracy and that the cheap availability and also executable in a low resource setting makes it very challenging. So it has to be cost effective. Let's see, what are the testing approaches or the diagnostic approaches that are present worldwide right now? The first type is the molecular test, which is based on determining or detecting the RNA particles that I showed you in the anatomy. So those RNA particles or the nucleic acid particles present inside the virus's body can be detected. That's a very confirmatory test, but as I'll show in the next slide, it has a limitation that it is only possible to detect the active infections. So after the infection, the first round of infection is gone, then there is usually no possibility of detecting the viral RNA after a few days. And also the collection method is usually from the nasopharyngeal swab. Sometimes, since this is a contagious disease, when the healthcare workers are trying to collect that with a swab, it generates a response of a reflex of sneezing, exposing the healthcare workers and other people present there for the collection. The antibody test, which detects the antibodies, which are the protein particles produced by the host or the human immune system against the virus antigens. We detect those. That's the other kind of test. In that, usually we use blood, which can be done by a venipuncture, that is a syringe collection. But usually we have come up with other easier methods like a finger prick or a dried spot of blood to do the rapid tests. And the third and the newest method, which is being applied for coronavirus tests, is the antigen test. This is the newest of the three testing types. It detects not the RNA particles, but the protein particles present in the virus. It can be usually the spike protein. It can also be the particles of the nucleocapsid protein N. So as I said, all of these tests definitely have limitations and positive aspects. But what we have to do, the challenge for the regulatory bodies and the scientists, is to come up with, say, a combinatorial approach or a practical approach where you would use these tests in a very pragmatic way or in a clinically well-advised way as a first line of screening, as a second line or confirmatory screening, so that in the least amount of time and with the least money spent, we can diagnose, screen, and do surveillance on most amount of people or a large population. That being said, antibody test is the only kind of test, although it has the limitation, let me first mention that, although it has the limitation that it cannot detect very early infections. So the antibodies, as we have seen, as it has been reported, are produced by the host immune system after a few days. Usually about 7 days, the first antibody starts being produced, the IgM, then IgG, which persists in blood for a longer time, usually comes up, say, around 10 to 14 days, but it persists longer for several weeks to a few months, as it has been shown, not a year as we were anticipating before. So that's the limitation for early days. It can do a false negative when the person is already infected. But the advantage of the antibody test is that, and the huge advantage is that it can detect past infections. Antigen test, as I said, it detects the protein particles of the virus as opposed to molecular test, which detects the amplified or the replicated the viral RNA or the viral nucleic acids, but both of these can only detect the active infections. Antigen test has an advantage over the molecular test that it can be done in very short time, just like the rapid antibody tests. But the molecular tests, once again, has a huge advantage over the others, being that it's accurate as it detects, and it's known as the gold standard universally for detecting these kinds of virus. Once again, as I said, in a pandemic situation, we cannot always resort to the most sophisticated and the gold standard method, but we have to come up with practical approaches to do most amount of diagnosis in the least amount of time with the most accuracy possible. The masked testings across countries, this is a graph from our world in data. This, as I said, the coronavirus statistics and the data keeps evolving. So I just collected this graph to show you the trend, which has not changed over the past 1 or 1.5 months. This was from the end of May. It shows that the United States, Italy, and the United Kingdom, they're doing a lot more tests than countries like India and Brazil. In India, I can talk about the comparison between United States and India, that probably mass testings going on at a larger scale or many more testings, that also leads to more detections. So not knowing the exact number of infected people, let's not be very complacent, that less percentages of people are infected and the virus is not going to run havoc in a particular country, like maybe India or Brazil. So mass testings are very important. Now, how can we make these testings using whatever I was talking about, the nucleic acid approach, the antibody or the antigen approach? I'm going to talk about how can we make this sustainable. Okay, sustainability is very important. Your own sustainability, our own social and mental sustainability, and also a sustainable approach for screening and surveillance of this pandemic and preparing for the next waves, so that we know the actual number of infections and detect them properly. So recently, as Dr. Shuman Das was also introducing me, then he mentioned that I'm involved in a project for coming up with these modified diagnostics to be applied in developing and specifically the resource-limited countries. So we came up, this was actually an existing method, the loop-mediated amplification, or the RT-LAMP. So we think that application of these in a proper way and integration with the software can revolutionize the diagnostics of COVID-19, definitely. So what are the advantages or differences of this method as opposed to or as compared to RT-PCR? Let me just, the figures are self-explanatory, specifically the one on the left, but let me just touch on that RT-LAMP, as opposed to RT-PCR, actually uses some loop primers. The students who are from molecular biology or biochemistry, or they have done a little bit of cytogenetics, you all know that for RT-PCR, or for amplification of the nucleic acid, we need primers, right? The nucleotide sequences. So what this RT-LAMP method does is it includes one or two sets of extra loop primers for the loop regions of those RNAs, which makes the detection specific at an isothermal temperature, which is very important. A very big factor in RT-PCR or real-time PCR, which is the sophisticated method but gold standard for detection, is that it needs different temperature for its annealing, amplification, etc. I'm not again going into all those jargons, but as you know, to detect the nucleic acid, we need a very sophisticated machine, an expensive machine, a skilled personnel for which we can look at the readout or the endpoint. It has to go to different temperatures at given times to give us the product for the readout. The difference of RT-LAMP or advantage is that it can be done isothermally, that is at a single temperature, usually between 60 to 65 degrees centigrade. It takes less than 1 hour to get the results at a constant temperature, remember? Whereas RT-PCR results are usually 2 to 2.5 hours at minimum. Sometimes, depending on the primers and the cycle, it can go up to 4 hours. Again, which makes it very difficult when you want to diagnose a lot of people or screen a lot of people. So if you use this RT-LAMP method and combine it with strips, like you put a sample on a strip and then do a heat treatment in a particular heat chamber, then you can do a readout using the color change. You can have dried LAMP reagents. We have come up and reviewed a lot of papers and researches going on. We can have dried reagents on the Eppendorf tubes itself and then put the sample inside so that we can again have a readout of the result very easily. And once again, at isothermal temperature, which means at 60 to 65 degrees centigrade. So it can be done at low resource or resource limited laboratories with very limited skills of the personnel, and the cost comes down a lot because we do not need freezing of the reagent. There are no limitations in storage and handling. So this method, only with a water bath, which can go up to 60-65 degree centigrade temperature, or a heat treatment box, or just a pot which has its temperature reason. This can be deployed even in the remote areas, the villages, countries like India and Brazil. And if there can be some testing sites set up where this can be done on a chip, a lab on a chip, and that data can be integrated with a software that is can be put into the computer system, in the integrated system, then storing the data and comparing it with the worldwide data becomes very easy, even just with an internet connection, a software and a laptop and a lab on a chip system. The other sustainable approach that I'm going to talk about for mass diagnosis is definitely based on antibody. Now, if you look at this graph, see that as I was talking about, the graph, the antibody expression, it does not start at the very onset of symptom. It takes a few days. You see a window period where after around 7 days or so, usually sometimes as early as 3 days, IgM becomes detectable, then IgG production begins. When the patient starts to recover, usually the IgM disappears, but the IgG remains in blood. And this graph here, which I collected from 1 month back or something, it says IgG remains in blood and provides long-term immunity. But from very recent Nature papers, we see that there are patient cohorts who have been affected by COVID-19, but their immunity goes down, or the IgG amount goes down in even 2 to 3 months. So let's not talk about long-term immunity. If a negative but a very useful finding that has come out of the screening and surveillance studies, like the one that I was involved in USA, which was the first large mass antibody testing study led by Stanford University, Dr. Aaron Ben-David. What we know from these kinds of studies run the world over now is that the concept of immunity passport is utopic. The human race has not developed any herd immunity as well. Herd immunity meaning at least 60 to 70% of the population is immune to this virus. It has not developed. So possibly our only legitimate chance of gaining a herd immunity is through vaccination. But again, I'll come to that at the end of my slides. Once again, I do not want to go into the nitty-gritty, but what happens is when we use the antibody testing, it can be done via ELISA. That is an enzyme-linked immunosorbent assay, which again requires instruments and skilled laboratory personnel, and also is time-consuming, has a very definite protocol. To avoid that, we can come up with a very rapid strip-based method, which has been done the world over by several companies, but we are still waiting on accuracy and specificity of these tests. But that is the very important sustainable approach that can be used for long-term screening and surveillance. Gold nanoparticles and conjugation of those gold nanoparticles into these nitrocellulose membranes or these adhesive strips of the rapid antibody testings can once again revolutionize these kinds of tests and make them very accurate and specific. So on the left, you can see just a picture from the Stanford study, which I was a part of. There are multiple serosurveys being carried on in the US, in European countries, in Brazil, and in India, as I know of till date. So what are serosurveys? What is serology, actually? So it's the study of serum or other kinds of body fluids. And effectively, it's the study of the antibodies present in the blood, which are produced in response to the SARS-CoV-2 virus. So studying a mass population for the antibodies, the percentages of antibodies, the types of antibodies, and the amounts of antibodies present, or sometimes just qualitatively, whether it's present or absent. Why does it help? First of all, that helps in surveillance and diagnostics. Second, that helps in attaining the correct pandemic statistics. Once again, if we go back and think about the antigen and the molecular test I was mentioning about, they can only detect active infections. And antibody testing that goes back and it can detect the past infections, at least for several weeks to say a month or two. That gives us a bigger and a better window to us to detect all the infections, which it's impossible to catch at the active phase. Within 9 to 10 days, or after 9 to 10 days of the initial infection, in most cases, the viral particles like the RNAs start to decline. So it's very difficult to catch all the infected individuals within that phase. That's why the importance of antibody testing and mass testings overall. There are some very few applauded, I won't say few, there are many studies going on, but few of them have been applauded because of the research design, because we have to apply these testings very effectively. Again, as I was saying, in conjunction with the clinical accuracy, the right kind of questionnaire, whether those people came in contact with infected people, what are their daily habits, and so on. What are the chances that they can be positive or negative? Combined with those questionnaires, we can arrive at a statistics of the disease and a better epidemiological modeling. That can definitely help in reshaping the public policies, like when to reopen, how much to reopen, and what depending on the infectivity and the population who are infected, how many or what percentage of people should be called back to work, whether it's safe for the healthcare workers. Once again, we are not anymore talking about immunity passports, because that is utopic. But definitely, we will have to sustain, we will have to reopen the economy and carry on things, not like it was before late 2019, but definitely with a new normal and adjust to that. So for reshaping those policies, knowing the correct statistics of the disease is definitely very important. Although this is not directly the topic of my today's presentation, but I would like to touch on these because it's an immediate effective approach, which is the convalescent plasma therapy. It's very much, intertwined with antibody research and serosurvey research. When you can detect the number of the percentage of population or the potential donors, who have been infected and who have generated antibodies against the SARS-CoV-2 virus, you can draw their purified sera and you could inject the severe patients for therapeutic work. And we have seen the world over, this has produced effects. This is one such report from a very important study. On the left, you can see how a lungs looks in a CT scan. The lung damages look when they have some very contagious virus infection, when it is acute pneumonia-like. You can see those opaque portions, which are called the ground glass opacities, that are usually basal in case of, and also these bright spots, which are the fluid accumulations that happens in case of SARS-CoV-2 infection. So in case of therapy, the median time, this was a study where 10 severe hospitalized patients were given convalescent plasma therapy. The median time was 16.5 days, and most showed improvement in 3 days. 7 patients even recovered from viremia, meaning that they didn't show any presence of virus particles or antigens inside of their body or their system. So we see, these are just 2 random patients that I displayed here, the scans from them, that these ground glass opacities and the fluid accumulations, they declined and the disease symptoms were alleviated significantly with convalescent plasma therapy. This was also another severe patient whom you see these opacities have been disturbed here. So the other sustainable approach is monoclonal antibody research and therapy. We see that by convalescent plasma therapy, actually, that's a concoction. There are several other protein particles present in the body which are injected into another severely affected or infected patient's blood that can generate sometimes some adverse reactions. So in this scenario, the sustainable approach is actually doing some monoclonal antibody research, synthesizing those and injecting a cocktail into the severe patient's body. There are challenges in that aspect as well, that there could be false negatives in early infection. So there can be low and moderate accuracy of the strips because we do not have universal approval of any one kind of rapid testing method, but people around research institutes and companies are working towards that. They have understood what's the importance of coming up with an accurate and specific strip-based or rapid testing method as opposed to a sophisticated one. The one disadvantage is that sometimes the antibody production is not so good in immunosuppressed individuals. That's why the importance of vaccination. IgM and IgG titer timelines also vary, and we do not know. We know for sure that there is no long-term immunity, but know that this antibody research and this serosurveys and this surveillance gives us this important information that even when we do the vaccines come in the market, it might not be just one-time vaccination and you are immune to everything. It might be seasonal, like they do with the flu vaccines, just the normal flu in the US. Or it might have to be some booster doses. Research is still going on in that. With that, I move to the other aspect of my presentation, which is facing the new normal together. This was all about sustainable diagnosis and screening and how we know that the first line of approach can be a rapid antibody test. A combinatorial approach can also be there, that is we can have a strip-based antigen and antibody test combined together, and they can be, if companies can come together and produce them at a very accessible or affordable rate, then that can also be a home-based or community-based diagnosis. Remember, you have to repeat those because they cannot be the early infections cannot be detected via the antibody method. So it has to be at least repeated weekly 2 to 3 times. If such a method could be come up with, then that could revolutionize the screening and the sustainable surveillance procedure. And then for the confirmed cases, they can go to the radiological imaging or to the nucleic acid method for confirmation. I think that gives the combinatorial approach, gives a very sustainable and affordable approach for pandemic preparedness for the next waves of this disease, and also for a sustainable screening right now. So as I said, it's very normal and natural. And we have faced this new normal together for a few months now. So all those negative thoughts and, there were reports coming in that some people who are not actually sick or who have not been, exposed to any kind of virus, they have also developed this psychological fear that, I have COVID, and that's affecting their mental health, that's affecting their immunity status, because as you know, your health, your physical strength is very much dependent on your mental health. So PTSD has been seen as a second tsunami of SARS-CoV-2 pandemic, but I think with a few months passing, people have adapted a little bit to this new normal, and have started gaining some sustainable approaches, some surveillance, screening, and behavioral changes. So for the behavioral changes at the social level, I would definitely talk about protecting the vulnerable. So we cannot be stigmatized. We have to have empathy towards people who have already, we know that they have been detected to be COVID positive. We have to be grateful for the service of the people, vendors and small business owners, including restaurants and others. They have to sustain the contract workers, the daily wagers, the essential workers like in food supply chain, in sanitation, pharmacists, banks, post offices, telecom, airline staff who are putting their lives in danger. We have to be grateful for the service of police, journalists, and be empathetic towards migrant workers and evacuees from other countries. The surveillance method that I was talking about, the antibody and antigen combinatorial screening method, the rapid strip-based testing, can be very effective in also the mass habitations like orphanages and old age homes, hostels and mess facilities, in slums with very poor resources, in prisons and in homeless shelters. So if we can achieve that and apply that with definitely organizational help and help from the government, we can get through this together. So what are the behavioral changes at the social level that we want to attend? These two are very great models. The left is an image, on the left is an image from Domino Park in New York, USA. As you can see, they have made chalked out some circles. People are out in the lawn of a park, but they're still social distancing and enjoying their day, sometimes with family, sometimes alone, but keeping the new norm, And on the right, this is actually a important image from a very model state who had been successful initially to control the pandemic effectively. That's from Kerala, where they were using umbrella to attend social distancing. So young people have to be, instead of sitting home and panicking, you have to come up with innovative methods and be a part of the conversation. Talk to your peers. These small innovations, these small behavioral changes can go a long way in saving your loved ones. Sometimes you think that we are immune to this because we are young. Now, research has shown that no, young people and healthy people are not immune to the virus. It affects the old and the young, rich and the poor, equally. So we have to do our part in preventing ourselves from getting affected and also protecting our loved ones. And this one is a known image. I do not need to explain any of this. There's wear a mask, wash your hands repeatedly, don't touch animals or livestock and then start eating. Cook your meals thoroughly. If you come in contact, if you have to care for elderly people or sick people, take the respective measures to protect yourself. When you are coughing or sneezing, cover your mouth and be mindful of your mental peace. Take regular sleep, eat healthy. You don't have to eat a lot of fancy things and go to the market to buy lots of vegetables every day. And that's how jeopardize your protection, but do it healthy and have regular sleep. So these are two important and interesting graphs I want to show. I'm almost towards the end of my presentation. Behavioral changes on the internet reflecting the adaptation, very, very important. It has shown some positives, coming out of this negative scenario. Different generations have increased distance learning and career planning in a very sustainable way. This behavior was not that much common before the pandemic. We were just running in a rat race, and we were used to go to our schools and colleges and universities, and we are now having to adapt to this digital age. But we do have a lot of time to spend with our families. We do have a lot of time to do our career planning and take a seat and relax, maybe play some board games and puzzles and talk to our loved ones. So a lot of planning, a lot of staying home and doing learning, engaging, enhancing your knowledge. That adaptability has come forth in the statistics. And I think that is what you all should also apply in your own life and take advantage of the situation to the fullest to sustain in this new normal. Once again, among the trending online behaviors, this shows that different generations, like the Gen Z, Gen X, and the boomers, they have increased traffic to the cooking sites. They're cooking and posting on social media and doing a lot of exercise as well, which is very important because your physical health and your mental health is very, very important rather than worrying about this pandemic. So in developing nations, we can use our people power to our advantage. We can do resource management and research. What are the means for that? Surveillance and diagnostics, which was the main part of my presentation. The scientific advancements are sustainable, and the scientific advancements are profound in this aspect. We have to mobilize the supplies and generate small response chains. India has shown that, before, I know that my parents used to go to the market to buy things, and now they're bringing those carts with fresh vegetables and sometimes fish and everything to you. So that's also paying the vendors well without any involvement of the middleman. Somehow it's boosting the economy at the grassroots level, I think, and that's the positive that's coming up. And also it's mobilizing supplies. So the important part, which seminars, webinars, or conferences like this are carrying out, are spreading awareness and proper knowledge and dispelling rumors. So there is a lot of clutter because this is everybody's problem now. So a lot of clutter. So use your own judgment and listen to the only clinical and the scientific truths, and be strong, be judgmental. But don't be judgmental of people, be judgmental of the facts and the rumors, definitely. So provide support to people who need, and in the long run, countries like India need to improve on their research and manufacturing. This negative aspect, the pandemic, has actually brought forward another positive thing. There is a bridge between the science and society, which was much needed. It's not something that is utopic, not something that people go to the labs, go to their classes and learn and then forget about it. It's not a 9-to-5 job. It affects our lives. So people have started taking interest, and that is very important in research and science. On the left, you can see the picture of the lady. I would actually like to see somebody say the name in the chat box. That's the lady who first took the electron micrograph images of coronavirus, and see her dedication. She didn't know that this would become a pandemic on this date. And that's how the advancements of the research started. And see, now we are here. At least we know what we know through the scientific research that's going on. And in the end, I want to say, this too shall pass. So hold tight and follow the rules, and just stay together, stay strong mentally, connect with your friends and family, and you would be able to pass through this. Let me stop sharing my screen.
Thank you, Dr. Das, for your illuminating, informative, and very useful lecture with some interesting diagrams and a different sort of view from California. And now I would like to request Dr. Chakraborty to flash some questions from the participants.
So I can see somebody asked, is there any cell-mediated immune response found? Yes, of course, that's a very good question. So there is T-cell mediated immune response found. And there are, as you know, there are different kinds of vaccination approaches going on. One is the nucleic acid mediated approach, which actually generates, and there are the live virus which carries this nucleic acid particles as well, like the Chadox vaccine in Oxford. So that actually does generate a T-cell mediated immunity. And all the SARS-CoV-2, when it enters the body, that does bifurcate into humoral and T-cell based immunity. There is. The only reason why the rapid tests are, stressing on the antigen-based immunity, because those particles are easy to detect in a strip-based method and not the T-cell-based one. But yeah, definitely there are that bifurcated response. Is there any other question? Yeah, trans-differentiation research definitely can be used, but for any treatment or not, that is a matter of research. But that's interesting, and I feel that yes, that can be of use definitely. That's all I'm going to say about that. Did I see another question? Okay. So I think Debadri, she has another last question, that it can produce secondary antibody against the primary one. If such can happen, can corticosteroids be given? Corticosteroids can be given. And if you remember during my presentation, I was mentioning about, so there is this phenomenon called ADME, which is the antibody initiated the overreaction when there is suboptimal neutralization. And when we use plasma therapy, then there are several factors or proteins in the patient's in the infected patients or the donor's blood, which can incite certain kind of reactions in the host as well. So this, including this, steroids can be given, and that has been done. And that is why the convalescent plasma therapy has only been used in case of very severe patients. And the sustainable approach in that case is coming up with monoclonal or synthetic antibody cocktails, which would not generate such kinds of antibody-induced responses or suboptimal neutralizations in the patient body. So yes, corticosteroids, but once again, sustainable is monoclonal synthetic antibody cocktail. That's both a scientific and right now a political question. So, but I don't want to say no comment. My thoughts are that Russia launching the corona vaccine at such a short time, there are mixed reactions to that. And we are not very sure unless we see the long-term effects in at least after they launch their first batch and 2 or 3 months have passed after that. So yes, they have skipped several important steps and rushed it. We are skeptical. The scientists around the world, they are skeptical about it. But once again, that's a scientific and political question. Let's keep our fingers crossed that somehow it works, because if it works, then it, it's advantageous for the whole world. So there's a question, vitamin C and vitamin D can prevent COVID infection. So vitamin C and vitamin D, they're dietary supplements which help as your immunity boosters. They cannot prevent, but it is important to have a vitamin C and D enriched diet, or rather just, go into the sun for a little bit so that your skin can synthesize vitamin D naturally, for a little bit, preventing your exposure to other people and maintaining social distancing. But know that these are not therapies, or these are not proven chemical preventatives for SARS-CoV-2, but they definitely are your immunity boosters. So remember to keep them in your diet and go out in the sun a little. Any other question?
No more question. That's all, ma'am.
Okay.
Very nice presentation. Thank you.
Yeah, thank you very much. It was a pleasure. Thanks.
Thank you, Dr. Das, for giving us the time at such odd hours.
No, that's fine. It's early morning. And in fact, I congratulate you all for such a nice, welcoming video. It made my morning. Thank you. Thank you all.
Thank you. Yeah. Thank you. Over to Dr. Chakraborty to proceed with the next session.
Okay. Welcome, Dr. Ratnadeep Ganguly. I take this opportunity to introduce Dr. Ratnadeep Ganguly, who is a brilliant scholar and won National Scholarship in 10th and 12th standard and ranked first in Joint Entrance Examination in 1994 among approximately 30,000 candidates. He passed MBBS examination in first division from Medical College, Calcutta in the year 1999 and completed MD Pathology in 2005 from SCB Medical College and Hospital, Kotak. He is a diplomat of National Board Pathology. He is a fellow of Royal College of Pathology, London, UK. He is a winner of Gordon Signey Fellowship, World Pathology Foundation for the academic year 2010 for GI and hepatobiliary pathology training, Massachusetts General Hospital, Harvard University. He worked in many reputed medical colleges in India and abroad. He was appointed as clinical observer in thoracic and pulmonary pathology in Medical College of Wisconsin, USA in 2010. In 2013, he was appointed as locum appointment for service specialist registrar in Queen Elizabeth II Lister Hospital, UK. He was specialty registrar at King's College Hospital, London, UK from 2014 to 2015. He was appointed as specialty doctor in histopathology in Colchester General Hospital, Essex, UK from 2015 to 2017 and presently appointed as consultant histopathologist in University Hospital, Coventry, Warwickshire, NHS Trust, Coventry, UK. He has several publications in peer-reviewed international and national journals. Welcome, Dr. Ratnadeep Ganguly. Over to Dr. Ganguly.
Hello, everyone. And am I being heard clearly?
Yeah.
I hope. Okay.
Absolutely.
Okay, thank you very much, Dr. Kaushiki Chakraborty, for that nice and humbling introduction. And thank you very much to the organizers for having me today. And thanks very much to the students for, I hope it's going to be a patient listening. I will try not to bore you. And so I will now go and share my screen. Just a second. Okay. So is my screen shared?
Absolutely.
Okay. Okay. Can you see the first page, right? Okay. That's
Yeah.
fine. So, yes. So, my discussion is from a doctor's perspective or a healthcare worker's perspective, broadly speaking, because that's what I am and that's what I have been working as all through my life. When I was initially invited to speak on this topic, my initial impulse was, oh, I don't know much about it, but let's read up. I did read up and I did a bit of research here and there. So here I am. So without further ado, let's go ahead. Can you see the first slide, everyone, right? Okay. So, the coronavirus disease, as is doing round in the world nowadays, is initially originated in Wuhan, a city in China, a pretty big city, and there was talk about that this was a leaked virus from the Wuhan Institute of Virology Laboratory, i.e., this was an artificially laboratory-made virus. But then there are other papers in Nature Medicine, pretty reputed papers. I hope you can get access to that. That's a free one. So that actually proved that the virus is a natural one, and the DNA sequencing is actually too random. There we are. It's a zoonotic virus originating in the bats, and first originated earlier this year in January. Also called SARS-CoV-2, also called human coronavirus 2019 or 19. It belongs to the family of viruses called coronaviridae, and it's a single-strand adenovirus of positive sense, and belonging to the same group of viruses or the family of viruses are other groups of other viruses like the original SARS-CoV, which did rounds in the world in the earlier part of this century, as well as the Middle Eastern respiratory syndrome coronavirus and some other less pathogenic viruses like HKU1 and so on and so forth. From the natural reservoir, the bats, it has to jump a few species to gain entry into the humans. So it is not a very natural process, but it has gained entry nonetheless for whatever way, we don't know. But there we are. It's with us at the moment. So the initial few slides are dedicated to be a pretty basic discussion about the disease. I hope many of you will be aware of that. In fact, most of you will be aware of that. But I'll go through them because without that, the discussion will not really get complete. And then I will gradually go towards the more important part of the discussion, which is the sustainability aspect. So most people get a mind flu-like illness. In many, it could be asymptomatic. The important thing is that they could transmit. It can make some people very severely ill. Usually the people who are elderly and with pre-existing comorbidities like asthma, COPDs, and cardiovascular conditions are more susceptible to develop a more severe disease, and eventually some of them may die. And the elderly and those with comorbidities have definitely a higher likelihood of death. Now, the issue with this coronavirus is not the case fatality or the mortality issues. The issue is with the transmission because it is so very highly infective. So let's discuss about the transmission. The transmission is through the respiratory route, usually through nose and mouth. It happens mainly through three ways. If you go to the WHO website, you can go into the details of this, but it's just an outline. So it can go through the droplet infections, which are slightly larger particles of 5 to 10 microns in size, which are emitted by every one of us when we cough or sneeze. And they usually reach up to 1 meter from the person who is coughing or sneezing. So these are called droplet infections, larger droplets. These don't remain suspended in air for long. Then you have the aerosols or the droplet nuclei, which are smaller particles, less than 5 microns in size. And these are given out as we speak and breathe, and also cough and sneeze. These remain airborne for much longer, and these travel for much longer than 1 meter. And these were initially debated as whether they can really transmit the coronavirus, but it has now been proven, or at least we are going towards proving it, that these can also transmit the virus. And these are also generated in multiple aerosol generating procedures, which mainly the healthcare workers are subjected to, like nebulization and ventilation and so on and so forth. And then the third way is through fomites, or things which are touched by the people who are infected, and with their contaminated hand. And then you touch that, from that again you touch your face or your nose or your mouth, and you get infection. And very importantly, asymptomatic carriers can transmit very effectively through all these means. So we don't have a vaccine at present. There are multiple candidates under trial, but the best way to prevent it is to avoid getting exposed to the virus. You have to wash hands regularly with soap water or 60% alcohol containing sanitizer. Normally 2 meter social distancing is advised, but with aerosol transmission becoming a distinct possibility, this may require a higher level of protection, longer social distancing, or wearing barriers like masks. And then, of course, you are not permitted to touch your mouth, nose, or eyes. These are the main ports of entry. You have to wash your hands for at least 20 seconds with either soap and water or hand sanitizer containing 60% or more alcohol. This is to be done after any potential contamination like nose blowing, sneezing, coughing, and there are a host of other things, to even touching animals, pets. And this is also to be done before eating and before preparing food, before touching your face for whatever reason. You are not supposed to touch anywhere in the face with unwashed or unsanitized hands, and all hand washing steps are to be followed. So you disinfect or clean both surfaces of the hand, the dorsum and the palm. You disinfect all fingers, the space between the fingers, the thumb, the nail, the wrist, and for healthcare workers, you also disinfect up to the elbow. So these are very important things, pretty basic, but very important. And then coming to social distancing and barriers inside the house, with any sick people or anyone with any chance of any sign of infection, at least 2 meter distance, wear masks. Outside, you don't know the status of the person you are coming close to, so maintain a 2 meter distance from people regardless of their infective status. Vulnerable people better stay at home, and social distancing, if that is not possible, then you can wear masks. There are different kinds of masks available nowadays. The N95 and the FFP2 and 3 masks are the most useful ones, because they prevent the transmission of the infection as much as possible. Optimum stoppage of transmission is done through them. The surgical masks are also good. They are slightly worse than the N95s though, and they are actually better than the homemade cloth masks. But all of them are better than wearing nothing on the face, because as a matter of common sense, that every mask is a kind of a barrier. So be careful about your masks, sterilize them, wash them, and use them. And masks with bulbs are not recommended, because they encourage a one-way transmission of air from the wearer to the outside. So these are not intended for protecting the society, people around you. It is only intended to protect the person who is personally wearing the mask. So those masks are not really recommended for stoppage of transmission. Then certain self-care protocols or methods we need to be following. We need to cover our faces while we sneeze or cough with elbow, not with the hand. If we use tissues, we are supposed to bin them immediately, and then we immediately hand wash or hand sanitize. The frequently touched surfaces that are there everywhere, like the knobs of the door, the buttons of lifts, the handrails next to the stairs, if we touch them, then these are, I mean, you have to wash your hands, and as much as possible, these surfaces need to be sterilized with soap water and alcohol as much as possible. Everyone needs to be on alert for symptoms, any temperature, any breathlessness, cough, dry cough especially, or even moist cough, anything. And then vulnerable people need also to be monitored through pulse oximetry as much as possible. So, in short, whatever we have discussed till now are the basic tenets of the so-called thing, the so-called lockdown thing that is going round in the world these days. So, what happens if we don't do the lockdown, don't follow the lockdown? You get a bell-shaped curve. The epidemic attains a peak very fast. The slope is quite steep and then comes down in an equal steep manner. But the problem is this period when the epidemic has really overwhelmed your healthcare system's capacity to deal with cases. So the idea or the purpose of instituting the lockdown at the correct time is to reduce this slope, make it a more gradual rise because the epidemic is going to rise anyway. That's a normal or natural process. So you make sure that the rise is gradual and the slope attained, the peak attained is less than what your healthcare can deal with. So the healthcare system is not overwhelmed, and then it goes down gradually, but then it is also prolonged. So your healthcare has to deal with the disease for a much longer period. But at least you have made sure that the healthcare is not overwhelmed. So that's very important. So let's see when exactly to impose lockdown. In different countries, we have got different experiences. Overall, it is usually right to impose the lockdown when the epidemic is starting to gain momentum. It's starting to ascend. The slope is really picking up. So if you impose the lockdown, you still get a peak. You get a gradual sort of slowing, and then you unlock when you think you are kind of in control of the disease. It keeps going down. Similar here in Germany, and similar here in Italy, and similar here in UK. You might argue in the UK they have released the lockdown slightly earlier, but it didn't really matter that much. Maybe there were some hitches here. Maybe there was some slowdown in the way the epidemic sort of went away, but it did work that way. However, in India, what we have seen is that the lockdown was instituted quite early, very early. That has resulted in a very gradual rise, very gradual rise. It's so gradual. In fact, all these graphs are drawn to scale. They are comparable to each other. So you can see how gradual. So the Indian curve is that prolonged. It's spanning the entire screen. So it's very gradual rise. But then at the point the epidemic is gradually gaining momentum, because the sustainability of the lockdown is an issue. You can't sustain the lockdown for very long because of the business and the social issues, social concerns. So ultimately you have to release. So the problem with that approach is that you have to release the lockdown at a stage when the epidemic is really gaining momentum. So, well, that has been the experience. We'll come to what exactly happened after these at a later slide. So just a brief discussion about the treatment of coronavirus disease. So the mainstay is oxygen therapy with positive pressure ventilation, either through CPAP, which is continuous positive airway pressure ventilation, or ECMO. ECMO is extracorporeal membrane oxygenation, where the body blood is made to pass through a series of membranes where it is artificially oxygenated outside the body. And that's assuming your own lungs are not able to oxygenate it that well. And then the last resort is ventilation, which is gradually falling to disrepute now. And then maintaining your fluid balance, that's very important as well. The oxygen saturation measured by pulse oximetry is the way to monitor the level of progress or the level of degeneration that is happening in the patient. And then there are many antivirals and other medications, some of them experimental, that are being used and have been used for treatment, like the antivirals like remdesivir, and then some antiparasitic drugs like ivermectin, monoclonal antibodies like tocilizumab, then antimalarial like hydroxychloroquine, then other antiviral drugs like interferon 1 beta and lopinavir, ritonavir, things like that. And also long-acting steroids like dexamethasone in very critical patients. They are giving good responses because they reduce the immune response, which is going against the body. We will discuss that later on. The treatment is to be done in isolation. There is no face-to-face contact with the family. The patient is not permitted to see his or her family members. The family members are not supposed to see the patient face-to-face, which is really the stressful part. Then a little discussion about the pathophysiology of the disease. It has evolved over the course of the epidemic in the society. Initially, you get a viral illness in the upper respiratory tract, but then it gradually gains into the lower respiratory tract, enters the lung, and through the type 2 pneumocytes and the endothelial cells, which have the specific receptors, which are the ACE2 receptors or angiotensin converting enzyme 2 receptors, it gains entry to those cells. So the type 2 pneumocytes and the endothelial cells are the main reservoir of the virus inside the body. So it attacks the blood vessels in the endothelial cells. And once the blood vessels are attacked, the endothelial cells are uneven, they degenerate, they die, and the patient undergoes through thromboembolic manifestations or conditions. The lungs are affected as a result of that. Initially, it was thought that it is a viral disease, just like flu, and then you get a secondary pneumonia, just like you get in many in many influenza virus diseases. You end up with a secondary pneumonia. But then in this case, it is the thromboembolic condition that is causing the pneumonitis. So that means that in addition to the lungs, there are multiple other organs that are also infected, and the patient may end up with the so-called multi-organ failure. The patient may end up with renal failure, the patient may end up with stroke-like symptoms in the brain, and multiple organs may get affected. And once the patient gets multi-organ failure, usually the prognosis is much worse from that stage. Very important in this disease as well, the so-called cytokine storm phenomenon. When the cytokine response in the body, the antibody response in the body goes overboard and attacks the body's own defenses, the body's own system. And that's when the effect of steroid is important to suppress from going overboard. And long-acting steroids like dexamethasone are being used for this purpose. So here we see the spike protein of the coronaviruses. They gain entry into the endothelial cell and type 2 pneumocytes through the ACE2 or angiotensin converting enzyme 2 receptors. And then this cell gets infected. Potentially, if you stop this joining by anti-ACE2 antibodies, you could have stopped the entry of the cells, something which is a subject of research. So what happens to the initial, so the immune response the patient goes through? This has been discussed in the previous lecture as well, but just a short briefing here. So most patients get an IgG as well as an IgM response. And if you consider both IgM and IgG response, you get 100% response by about 19 days, either IgG or IgM. And both IgM and IgG responses plateau after about 6 days from seroconversion. I would highly recommend you go through this paper. This is freely available, and you can gain many insights into the way the immune response or the antibody response progresses in these patients. So the serological diagnosis criteria, as recommended by the WHO, in this disease are very similar to those used for the MERS coronavirus or the Middle Eastern Respiratory Syndrome coronavirus disease infection. So first of all, you need to get a seroconversion, which means the person becoming negative, who was negative becoming positive, either that or a person who was already positive at low positive level gains a 4-fold increase in the IgG specific antibody titers. So these are both necessary and sufficient for the serological diagnosis of COVID-19. These are used as useful adjuncts or useful complements to the RT-PCR-based viral RNA detection. And these are actually sensitive and confirm very early coronavirus infections. The nucleic acid tests are more expensive. In fact, the antibody tests are far less expensive than that, and these can be done at a much faster level, much speedier level. So this is also useful for screening large populations, asymptomatic patients, and things like that. So confirming suspected coronavirus 19 cases as early as possible with the help of the serological testing would reduce exposure risk during the repeated sampling, and this will save the valuable RT-PCR and keep them in reserve for the more severe conditions or more in situations where the resources are available only. So the serological tests can be used as a very useful adjunct and complement to this. You can go through this paper, which will give a lot of insights into this the development of antibody responses, as well as how the serological tests go ahead. So we'll come to the vaccine development phase now. Now, there are a number of candidate vaccines, a lot of candidate vaccines being researched, being tested in different parts of the world now. The basics of vaccine research is that the vaccine has to go through a phase 1 and a phase 2 and a phase 3 of trials. In the phase 1, there are very small groups of people who are allowed to receive the trial vaccine. It is just a beginning. And in phase 2, the study and the trial is expanded. It is given to a set of people who has got similar characteristics in terms of age and physical health, similar to those who are going to be the target population. And in phase 3, it is further expanded and it is evaluated for efficacy and safety. And mind you, in certain vaccines, you do need a phase 4, a more formal safety and efficacy study. And then there are many potential vaccine targets being evaluated for coronaviruses. You can either use the inactivated virus, you can use the live attenuated virus, you can use a part of the virus, which is the protein subunit, you can use some virus-like particles, you can use non-replicating or replicating virus vectors and viral nucleic acids, or with the RNA or DNA. And all of these have their pros and cons. Like the inactivated virus, it is easy to make a vaccine like that, but then it produces a weaker immune response. But the good thing is that it can be also given to people with weakened immune status. The live attenuated virus, on the other hand, produces a very strong immune response. It is more like a real disease, but it is not suitable for immunosuppressed individuals, like people who have received organ transplants, people who are on renal failure, the people who are on who are having HIV simultaneously. They cannot be given live attenuated virus vaccines. You can try the protein subunit vaccines, which are easy to produce, but then they also give a weaker immune response. And then virus-like particles, they give a stronger response than the protein subunit vaccines, but there are issues with stability, purification, and quantification, etc. Then non-replicating and replicating virus vectors are better than the segment vaccines. They are better than the protein subunit vaccines, but there are issues with preservation and production. And then specifically in case of the non-replicating virus vector, the usual vector is adenovirus or some other fudge virus, things like that. Some people may be already infected by those viruses, so they have a natural immunity or antibody titer already produced in their bodies against those viral vectors. So the non-replicating virus vector the vaccine will not work in those patients. And then the RNA and the DNA vaccines, they are easier to make, but they have some preservation issues, and they do require adjuvants, especially the DNA vaccines. You can just Google vaccine tracker. There's a nice vaccine tracker in the WHO website, and there is one at the previous reference that I gave you. So you can find out a lot more about vaccines, the particular candidates, their pros and cons, and which one is going to be successful, which one is not going to be successful, the predictions and projections and things like that. So these are just a few of the vaccines that are the forerunners in the present scenario. The most important is, I would say, the Chadox vaccine being developed by University of Oxford and AstraZeneca. And this is a vaccine that is using the non-replicating viral vector that is as the platform. And this has entered the phase 3 trials, and it is likely to come to the market, hopefully, in the coming months. And then there are 3 Chinese vaccines, which are all using inactivated virus. These are also entering phase 3. And then there are 2 vaccines being produced or tried in India. One is the Bharat Biotech vaccine, which is the inactivated virus vaccine, and then a DNA vaccine being produced by Cadila. These are both in phase 1 and 2. And there are some other nucleic acid vaccines or RNA-based vaccines, one of them being produced in the Imperial College London. And there are 2 other vaccines which are also nucleic acid-based vaccines being promoted by the American government. One of them, the Moderna vaccine, has entered or is almost entering phase 3. So if you go to the WHO website, you can find out a lot more about vaccines. You can get a more complete list. And there are approximately, if I remember correctly, about 28 or 30 vaccines. So that's obviously beyond the scope of this discussion. So I'll come to what is exactly happening now. Now, the previous graph that I had shown you actually shows what has happened in the few months, say till June or early July. And this is what's happening now. So after that initial peak, that has receded, that has come down, and then some days of a lull, and then some countries are going towards a second peak. You see that little rise again, which is very prominent in case of the US, and it is also prominent in case of this country, Spain. So many countries are actually going through the second peak. Fortunately, the mortality in the second peak has been much lower than the first peak, but fingers crossed at the moment, and we need to wait and see what exactly is going to happen in the coming days. Because remember, the mortality usually lags behind the causation of infection by usually 14 days or so. So we don't really know what exactly is in store for us. So the mortality that we are seeing today is actually from the infections 14 days back. So the disease is here going to stay. It is is going to be with us for a while. It is going to be with us till a vaccine is found, at least. And what is going to happen even after that? There have been other examples of vaccine preventable diseases, like the avian influenza, the so-called bird flu, the SARS, the initial SARS. All these have remained in the society. They have remained in the society at a much lower level, much less important level in terms of public health, but they have remained in the society. They have not been wiped out or eradicated. So in other words, they become endemic from being epidemic. And the low intensity community transmission at a slightly higher level is also possible, like what happens in case of malaria in Africa, or even some Asian countries, Southeast Asian countries mainly. That could also happen. The virus could remain in the society at for a much longer period at a much lesser level. So it's going to be some years before we can actually eradicate this virus, just like we have eradicated polio and smallpox. So the issues regarding sustainability are manifold. There are social issues, there are medical resource related issues, and there are economic issues. Now, I will confine myself to discussing mainly the social and the medical part of it, because I'm no economist, obviously. And the social sustainability is mainly as to deciding the policies, when exactly to impose the social distancing and the staying at home guidelines, when exactly to shut down or gradually shut down your economy. It has to be early enough, but not too early. And you have to also have a backup system in the form of contact tracing and tracking. So that's also very important. And then you have to determine how much your medical resources, how much your healthcare system is able to cope with. In spite of all the measures, the epidemic is definitely going to progress. And if you impose a lockdown at a very effective way, you are going to reduce the peak, but you're still going to have an epidemic which is going to attain some sort of peak. So you still need to define what sort of medical resources you have, what sort of level of epidemic you can cope with at its maximum, so as to see that your healthcare system does not get overwhelmed. That is very, very important to ensure, and that will ensure that unnecessary deaths are avoided. Many people are going to die because of underlying illnesses, elderly people, people with susceptible issues, conditions, etc. Some of them are going to die. You cannot avoid that with the best of your efforts, but you are going to avoid as much as possible. And then the economic sustainability is very important which is related to both social and medical issues, both. The society as a whole cannot sustain lockdown for too long. That's the most important thing in it. So once you impose the lockdown, it's a one-way process. You cannot come back so easily. You have to come back at some point, but you have to keep maintaining the lockdown for a while, and then you need to decide when exactly to impose the lockdown. People get psychologically exhausted from continuous fear and worry. Many people suffer from PTSD, post-traumatic stress disorders, depressions, and things like that. And then it just feels like that you are continuously under watch, you are continuously under curfew, your behavior, somebody is breathing down your neck all the time. So that's the social part of the sustainability thing, because the human behavior is mainly to socialize and mix with people. Even in the workplace, we do chat with our colleagues, we do exchange our views, there is some physical contact, there is always some chatting, some informal gatherings and things like that. You're going to miss on all that. So these are the things that affect people. Internet calls and video calls are here to help, but there is a limit to that. I'm sorry. So to some extent, we have seen that the working population also wants to be in the office, in the setting of colleague interaction to an extent. Although working from home has got its advantages, but it also has its disadvantages. So in fact, the studies have shown that many people nowadays would perhaps suggest a mix of home working and on-site working to be maintained to enhance the productivity. What are the social issues as regards the continuance of lockdown? So the very important issue, as I have already discussed, is when exactly to impose the lockdown. Not too early, not too late. Very important. And then the lockdown does slow down the spread of the disease. It so-called flattens the curve somewhat. The peak is lower, so as your healthcare system is not overwhelmed, and you can channelize your drugs, healthcare manpower and equipment, your hospitals, your beds, everything for the needy that way, and you can reduce the number of unwanted deaths. But it all comes with a very heavy cost, at a very heavy cost in terms of economy, in terms of loss of your GDP, in terms of having a debt-ridden economy on the verge of depression, and a long recovery period taking years. And then there are medical resource related susceptibility. You do flatten the curve. You reduce the maximum extent of the number of infective cases that you need to deal with at a time. But then there is also the caveat that you increase the duration of the epidemic that you are going to suffer from, the society is going to suffer from. So there are other diseases, other surgeries, other laboratory tests, everything else that is still happening in the society. The COVID hasn't reduced the incidence of the other things. So those things are going to happen, and those surgeries, those laboratory tests, for example, the prosthetic hip replacement, prosthetic knee replacement, very important for people who are suffering from arthritis, people who are suffering from fractures, etc., elderly people. Those take a backseat because a lot of the OTs in different hospitals are converted to wards to take care of the coronavirus patient, to deal with the increased number of the coronavirus patient. So your resources are going to be under a lot of stretch, under a lot of strain. And in fact, even the cancer surgeries have suffered in some areas of the world, though they are sort of semi-emergency. They are not fully emergency, just not like a heart attack or a stroke patient, but they still need to take place, but they have also suffered. So there is a buildup of backlog and then buildup of coronavirus cases. Overall, this becomes like a vicious cycle. And internet consultations with doctors do help, but this is not a full substitute because the doctors are not able to physically touch the patient. They are not able to auscultate the chest, for example, and listen to the chest. They are not able to touch your tummy and tell you the diagnosis. They are not able to do those. So these are important issues. But then I agree that internet does play a very positive role in at least mitigating some effects of the lockdown. And then if you discuss the virtual consultation with doctors, we still need a legal framework guiding that, because we still don't know how much the doctor is accountable to the patient as opposed to a physical face-to-face consultation. How much the doctor is accountable for typing errors, for the errors of the medicines being prescribed, for issues that are happening due to hearsay, for I'm telling you something and you're hearing something, things like that, that would easily happen through the internet. So those laws and those legal frameworks needs to be decided as well. And then there is the economic cost, effects of prolonged lockdown, the small businesses, the restaurants, the healthcare sectors, the hospitality sectors, they bear the brunt of this economic meltdown, the economic cost of that. And then there is exponential rise in medical care expenses in the form of all equipments, employing extra healthcare manpower, investing heavily into vaccine research and development, investing heavily into the pharma companies to develop more drugs for more medicines, or at least produce more medicines which are known medicines and produce more of them. So there is a lot of investment that way. And then the customer confidence in the society bottom lines, because people are simply too afraid to go out and shop. So unfortunately, however, it's not entirely the grim story or the bad story. The good thing is that we are going towards the so-called new normal, and some places have actually achieved or got a taste of the so-called new normal already. We have seen a spurt in the online shopping. We have seen a lot of virtual showrooms coming up. The home deliveries from restaurants and groceries and pharmacies have really improved in the recent months. People have come out, they have helped each other, and societies have come together like never before. And then we have come to that so-called new normal, a mix of socially distanced business, dining out and traveling, doing everything but maintaining the so-called social distancing. And a lot of things are being done over the internet. We are also getting less pollution in the air from less traffic, from less buses, less cars, more family time, and maybe also dealing with less amount of stress with working from home. And then household expenses may also go slightly down as well. So these are the positive aspects, and these are what we call the new normal. So that's it, and that's where I work. Only part-time these days, because most of the time I'm working from home. So thank you very much, and I would welcome any questions from your end.
Thank you, Dr. Ratnadeep Ganguly. Very nice and informative session. Well, just out of curiosity, I'd like to ask you one question. Am I audible to you? Hello?
Yes,
Yeah.
I'm fine.
Am I audible? No? Okay. Just out of curiosity, I would like to ask you one question. The vaccine you told about, almost all of them are attenuated viral vaccine, right?
Yes.
So I'd
Now,
like to
some of them
know.
are inactivated also. Yeah.
DNA vaccine, I mean about chimeric DNA vaccines. For example, if you take some more effective portion from SARS-CoV-2, and if it is attached with the effective portion of coronavirus 19, so how does it work? I mean, is there any research going on about that recombinant chimeric vaccine, DNA vaccine?
Well, yes, that's a very good question, and that's a very good idea, actually. The chimeric vaccines are usually from the virus. You take certain proteins and certain subunits from the viruses, and you put them in other viruses, which are usually the adenovirus vectors or the phage virus vectors, and then you produce a chimeric vaccine, and then that is used. But the problem with that approach is that the patient may already be immune or already having been subjected to the adenovirus infection. They may be already immune to that, so that patient will straight away reject that vaccine, so that patient will not have an immune response to the vaccine. The other thing is that whether any chimeric vaccine is produced with the present SARS-CoV-19 and the previously known SARS-CoV virus, which was the early 2001-2002 virus. Yes, I hear there are certain ones, but they are not part of this WHO website listed vaccines. I don't know why, but they are. I read about them a few days ago as well. They are going to come up, but I don't know why they are not registered. Maybe some of them have not been registered, just like the Russian vaccine, which has come up yesterday, 2 days before. That's also not registered. So people are a bit skeptical about those vaccines because of the way the researchers have adopted.
Okay. Thank you, Dr. Ganguly. Some questions are waiting for you.
Yeah, sure. Okay, so I can see one question. So is there a mutation of the spike protein on the new COVID virus due to rapid evolution, as the previous COVID virus can only affect bats? Yes, very possibly. As I said in the beginning of the discussion, that the virus has jumped species, because it is not likely that the bat-derived virus would normally affect humans. It has definitely jumped one or two species at least. So we don't really know there is any mutation that in the spike protein that has resulted in the jump or not. But then the virus, all the flu viruses in the world, they rapidly mutate, and that's the reason why you are supposed to get a flu vaccine the susceptible people, they are supposed to get flu vaccine every year, because the flu viruses keep mutating, and you need to cover the new strains that are coming up. So there's no reason why the coronavirus 19 itself will not mutate as well. And we just pray it stays in its present form for a while, so we can at least get our vaccines tested and get the result of our vaccines for a while. As of now, we already know there are about 6 strains of the virus doing round in different parts of the world. Already in India, in fact, there are 6 strains detected. So there are definitely other more viral strains detected all over the world. So yes, definitely mutation is a possibility.
Next question.
Okay, let's see that. Why is there so much controversy regarding the type of effective masks we still? Okay, so that's very important question. I was hoping someone will ask this. Now then, there are different kinds of masks available in the market. You can use a simple cloth as a barrier. That is also effective. That's not bad. And then there are the surgical masks, which are the 3-layered masks. Because if you use a surgical mask, you'll see that there are multiple folds, which sort of become flattened as you stretch the mask on your mouth and nose. So these are 3-layered masks. The 3-layer structure is supposed to protect you from entry and exit of virus. More exit than entry, because the person wearing the mask is still a bit susceptible to whatever is coming to him or her, because it is still not a fully blocking mask, because there are gaps between the nose and the mask, there are gaps between the ear and the mask, and there are gaps underneath in your chin, this area as well. So that mask, while it is more effective than the cloth mask, is still not a foolproof protection. It does protect against, however, the exit of the virus, because when it comes to exit of the virus, it gets more dispersed from the person to the person. So if you stop the flow of the virus from you, if you are an infected person, if you stop the flow of virus from you, it can only enter the atmosphere through those gaps that I was saying. Here, here, or here. These are the 3 places. So it is already diluted, and then it is going into the atmosphere, it is further diluted with flowing air. So there is much less likelihood of another person getting infected. And for the person who is wearing it, if the virus gains entry through these gaps, there is some chance of getting infected, yes. But it is, obviously, as you understand, this is simple common sense, that this is better than wearing nothing, better than using nothing. And then there are N95 masks, or FFP2 and 3 masks, which are sort of round and white in color usually, and they hold here snugly. There is much less gap. There is not much gap between here, etc. And the layers that they use are also much more effective in protection. They are much more effective in the transmission, preventing the transmission of viruses. So they offer better two-way protection. Even then, the entry of the virus is still more a possibility than the exit of the virus, because the more you use the mask, the more it becomes moist with your own breathed out air. It becomes moist, and the more moist it is, the more you get to smell things, and the more you smell the gases outside, like you're outside, you're smelling the smell of flowers, you're smelling the petrol, you're smelling everything. So once you start smelling things, you understand that something is going wrong, that things are gaining entry, that gases are gaining entry. So the virus can also gain entry if it is moist. So the water does act as a medium that facilitates the entry of the virus. So yes, there are a lot of types of masks, to summarize, and a lot of masks for different uses, and a lot of masks offering different levels of protection for the wearer and for the society. So that's all the controversy where it originates from.
Yeah. Another question.
Okay. Okay. So I understand that you mean that it has gone into community spreads. That's what you mean by stage 3. Is that correct? Because in UK, the stage 3 terminology is not used. I would take it that you mean that how do you say it has started? It has gone into stage 3, i.e., the community spread stage. The definition of community spread, as per WHO, is that when you get an infected person and you cannot identify the source of infection, or you cannot track and trace anymore, you cannot find out the person who has transmitted or got that person infected. So that's when you say there is community spread, when there is unknown source. So that's when it is supposed to enter stage 3, and that's when the infection really picks up. And that's when you are also beyond the containment phase. You are into the slowing down phase. You are actually imposing lockdown at that phase to slow down the spread of virus, to slow down the number of persons getting infected at a time, and you cannot contain anymore, because when you know the source of infection, you can still contain. You can still seclude the person, quarantine the person, and quarantine the source, and hope that no one else got infected. But if you cannot determine the source, then you are really in stage 3, and then you really need to slow down the infection. Okay, I have another
Next
question.
question.
Yeah. Does the COVID virus really cause blood clotting inside us? Can blood thinners be of any use? Yes. Yes, definitely. Because the pathology of the COVID virus, as I said, is that it has evolved over the progress of the epidemic. So now the main stress is on the endothelial damage that is being produced by the virus, mainly in the endothelial cells of the lung capillaries and elsewhere in the body, mainly in the lung. So once that phase is there, because it does take the virus about 6-7 days to attain that phase. Initially, in that stage, it was the upper respiratory tract, and then some lower respiratory tract infections symptoms take place. And then once the virus goes into the endothelial cells, you do get those thromboembolic conditions. You, because the endothelial cells die, and you need your blood to flow normally, smoothly through your capillaries, blood vessels, etc. For that, you need a surface on the blood vessel, a smooth surface on the blood vessel. So when the endothelial cells are damaged, you get an uneven surface. The underlying collagen is exposed. Once the underlying collagen is exposed, it induces the clotting mechanisms to start in the blood. It induces the fibrinogens to start getting active and to convert to fibrin. And that results in clotting, localized clottings initially, and then finally, once the localized clot is there, that makes the surface further uneven. The more uneven that is, the more clotting takes place. Not only fibrinogen, after the fibrin is produced, the platelets get clogged, entrapped inside that fibrin mesh, and that you end up with a clot. And once that clot is lodged in one place, that's fine. You get a manifestation in that organ. Once that clot travels from the lung, is called an embolus. So an embolus can travel from lung to, say, elsewhere, to the kidney to produce renal failure, to the brain to produce stroke, so on and so forth. And blood thinners, yes, they are definitely of use. Heparins are being used. The other tranexamic acids and other blood thinners are also being used. But these are early days. It is still not foolproof. Okay, as we speak, we have another question here.
Yeah.
Uh, can medical autopsy of lung from individuals who died due to SARS-CoV-19 be of any use? Yes, absolutely. That's a very good question, actually. You can actually see when you get the lungs from these patients, you can actually see two things. One is that you can see the manifestations of the pneumonia, the consolidation, the mucus and the mucin that is produced in the lung alveoli. They are clogging the lung air spaces and making the lung becoming solid. And that's called a hepatization of the lung. The lung appears to be like a liver, because the normal lung is actually like a sponge. It floats in the in water. From a dead person, if you see the lung, it will be like a sponge. If the person has died initially, I mean very recently, and then once the person has got pneumonitis or pneumonia, secondary to COVID or whatever else, that person will be having a very solidified lung. So that lung will not float in water. It will, go down into the water very quickly. And so that's the hepatization phase of the lung when you get consolidated. Like if you remember the CT scan pictures the previous speaker was showing, it was showing the consolidated lung picture in the basal region due to fluid accumulation. So this is one. And the second thing that you would get from in the lungs, in fact, from the lungs studies, we have understood the pathology now, is that the thromboembolic manifestations that I was referring to earlier, because you get to see the thromboembolic, the thrombi, the embolus, clogging the blood vessels and resulting in an infarct, resulting in an ischemia farther distal to the supply territory of that artery. Does that make sense? So you do get ischemic manifestations as a result of the thromboembolic things, and you do get consolidations as a result of the hepatization of the lung, which happens in pneumonias. You get these two things in COVID lungs, basically. Okay.
Another question.
Okay. Feces of an infected person. Okay. Why not? Well, theoretically possible, but this is not the main source of infection. The fecal-oral route of transmission, any body fluid, blood, urine, fecal matter, any body fluid is potentially infected. So that is treated as such in the hospital, in the places which are taking care of the patients. So these are also treated as such as biological wastes. But the problem is that the fecal-oral route of transmission, as happens in case of other bacteria, say like cholera, okay? So the bacteria has to go through the GI tract, which is a big defense. So in case of this, the virus also has to go through the stomach acids, which is the biggest defense here. So not many cases have been detected. A few anecdotal evidence of fecal-oral transmission have been there, but I would stay very careful. In fact, personally, anything given out from the patient is really infected. That's the reason why anyone dying on because of COVID is packed that way in plastic bags and all. So yes, it is a potential source. But then the respiratory source is so much more important that it does take a backseat. Okay, so the next question: mumps, measles, rubella vaccines are also recommended in treatment of COVID. I want to know what is the role of it. Does it really help? Okay. Now, MMR vaccine has been tried in few areas. I don't know the results have been very encouraging or not, because if at all there is some evidence of infection, then I mean some evidence of protection, then these are anecdotal, and this will require its own time to produce the antibody response or the T cell mediated immune response. So it's still early days. I would not jump into conclusions regarding that. These are anecdotal evidences, anecdotal things being tried everywhere in the world. So there's nothing proven yet.
Another question.
Okay. Who undergoes? Oh, sorry, it disappeared. Okay. Bilateral lung transplant followed by cancer due to postmortem addiction have higher mortality. Persons who undergoes bilateral lung transplant and followed by cancer due to post-transplant addiction have higher mortality. Yes, I mean, I assume that you are going to ask that the patient also has COVID and has these things that you mentioned, and then why this person has higher mortality. Okay. If you, first of all, if some patient is transplanted, any organ is transplanted, the patient has to be immunosuppressed. The patient has to be treated with a lot of powerful drugs like cyclosporin, azathioprine, and things like that, which will blunt the normal immune response, which would have otherwise rejected the graft. So if there is a lung transplantation or any other transplantation, there is a time that it takes for engraftment. There is a time when you have to artificially immunosuppress the patients, and you have to artificially keep the patients in a very secluded scenario where he or she is less exposed to infections. That is the first thing. If you are exposed to coronavirus during that phase, of course, you can imagine what will happen. Now, if there is a bilateral lung transplantation already there is some compromise in the function of the lung. There is the capacity of the lung is at least partially impaired. So if there is a partial impairment of the lung function, that patient has to be treated the way you will treat a COPD patient or an asthma patient with a compromised lung function already. So if you treat a patient that way, you can imagine that if a patient with compromised lung function by natural diseases versus that bilateral lung transplantation, they are going to behave in the same way because the lung capacity is limited. You are not going to achieve the full lung capacity of the normal lungs ever. So that's going to be an important thing, and immunosuppression as well. Inhalers containing budesonide are being used in treatment of the disease. If it is so, then people who use it, are they at low risk of developing this disease? Yeah, this is something I read about only 2 days ago. Budesonide and other steroids, these are steroid inhalers. And steroids are by nature immunosuppressant, we know. So immunosuppression is very important in this disease. In fact, judicious use of immunosuppression, I must say, because immunosuppression takes care of the so-called cytokine storm phase of the disease. When the body's own immune responses, the interferons, etc., they go out of control, and you need to blunt that response, making sure you maintain some level of immunity so that the virus itself doesn't go berserk and start attacking everyone. So yes, budesonide acts through the same mechanism because it is also a steroid container, and if you give through the inhalation route, it goes into the lung, it goes into the airways, and it should have some effect. Although it has not been used in large scale, the large scale use has been confined to dexamethasone and prednisolone. Oh, Sanjana, thanks for that.
No.
Okay, so in order to have a good sleep, kindly prescribe for me a good sleep. Okay. Don't get too tensed about coronavirus. This is going to stay with us for a while. This is not a sort of a notice that I'm giving. This is just the reality we are going to face. So let's not get overwhelmed by that. Let's not get overwhelmed by that. Let's continue our normal activities and get to what we call that new normal. So that's important. Lockdown is to slow down the community spread. Do you think 2 days lockdown is effective to us now? You are dragging me into uncharted territories. Now, okay, so, well, it will theoretically have some effect. If you think it will, if you have a 2-day lockdown, if people follow it religiously, properly, if you don't go out and if you see that other people are not going out, buses are not operating, transports are not operating, shops are not opening, everybody is staying home or working from home, or schools also not taking place, then people are effectively staying home. That has some effect in some sort of effect, some sort of temporary effect in slowing down the disease. Eventually, it is definitely not going to be as effective as a proper prolonged 2-week or 3-week lockdown. But definitely, yes, some effect will be there. But God knows how much. Those studies are not done. So there we are.
I think this is the last question.
All right. Can transmitted by hair. You are asking it to the wrong person. Okay. We are very aware of the time the virus can stay alive in various surfaces. What about hair or fur or pets? Depends on the type of surface you're talking about. Pets, hairs, and then clothings and papers, and then metallic surfaces. These all have different sort of viability range or timings for the virus. The metallic surfaces or the glass surfaces, as far as I am aware, has got a larger or longer period of viability for the virus. This has been said to be up to 7 days in some cases. Paper and clothings, on the other hand, are lower. They are usually, I don't know, whatever I have read is about 2 to 3 days. But then I would not go into those things and those speculations. I would always wash my clothes. If I come back from the hospital, I always wash my clothes and then wear it the next day. And I will take a good shower. So skin surface or any other clothings, papers, etc. Papers, well, I'll let it lie down in an air for a while before I handle it. So yes, that's how we prevent, without going into those speculations, because nobody knows really. And there will be controversies.
Thanks a lot, Dr. Ratnadev Ganguly, for
Thank you.
lending your valuable time for us and making us enriched. Thanks a lot.
Thank you very much. I really loved being here and enjoyed. And thank you very much. Hope someone is benefited from this. Thanks.
Well, I think this is the end of the session. I mean, technical session 1. Few people were asking about feedback link. Okay, you will be given feedback link tomorrow at the end of the technical session 2. Surely you'll be given feedback link. So don't worry about feedback link. Stay tuned with us. Tomorrow we will meet sharp at 6:30 p.m. But stay with us for a while today, because we are going to show you small audio-visual clippings where to salute our COVID warriors. So stay with us, stay tuned till tomorrow. Tomorrow we'll meet sharp at 6:30 p.m. for technical session 2. Good night.
End the session, please. End the session.